Danke, Christian. Und nun ist es mir eine große Ehre, den nächsten Keynote-Speaker kurz vorstellen zu dürfen. Es ist Paolo Dotto. At the same time, I will switch to English now, so he can rest assured I'm only saying positive things about you. His curriculum reads like a list of the best universities of this world. You did study medicine in Turin, then you moved to do a PhD in genetics at the Rockefeller. Postdoc at MIT, Cambridge. So, assistant professor in Yale as the next station, associate professor in Harvard, where I read you founded the laboratory for cutaneous biology, became a full professor in Harvard, and took on a position at Massachusetts General Hospital. So, as we all know, this is really one of the best institutions in this world for sure. And then since 2002, you actually kept your position in Massachusetts General and also moved to Lausanne University and are there in the Department of Biochemistry. And we all know that you are a real true key opinion leader in the field of the keratinocyte biology and especially cancerous transformation. And you have a very interesting topic of today, sexual dimorphism in cancer susceptibility. I'm really keen to learn what this is about. So the stage is yours. Please, Paolo, we are very happy to have you here and that you came from Lausanne. Thank you.
Thank you very much, Thomas. And thank you very much for the very kind invitation of the organizers. And so, as Thomas mentioned, I am an experimentalist, and I guess this talk to me is highly experimental because I actually never gave a sort of a virtual talk. Luckily, there is somebody in the audience, and I usually like to have very interactive presentations, but I'll try to make this talk as thought-provoking as possible. So I hope I will skip maybe on some of the hard data that can be found in some our publications. So, no.
Forward is with this one, and here's the laser pointer.
So wait a minute.
The top button is the laser pointer.
This one?
Yes.
Okay, good. And so, yes, for many years we have been focusing on skin cancer as a model for, in general, for squamous cell carcinoma development. And so the previous talk was a very nice introduction of the relevance of the system, not only in the skin, but obviously in many other internal organs. And so we've been focusing on the interconnection between transcription and epigenetic regulation, both in the epithelial compartment and in the stromal compartment, and then later also on the interplay between transcription DNA damage, and also on the idea that stromal cells can actually play a very important, sometimes primary role in triggering this cancer development. And so today I will briefly overview some of our work, but trying to put that in the context of sexual differences in cancer susceptibility, which I'm very fascinated as a topic. We're just starting to work on it, and it's very complex, but I think worth attention. Why about studying sex differences in cancer susceptibility? It's clear that over the last few years, there's been made tremendous success in treatment of patients, cancer treatment. But with this success also came an exponential increase in costs. And so the success obviously is leading to targeted therapies, as we just heard before, and also the idea of developing some kind of precision medicine. We are actually very much interested also in another very important dimension of this whole thing, which is actually cancer prevention. And actually, I just mentioned that we started a whole new PhD program in cancer prevention with funding by the European Union throughout Europe, and with a major focus on sex hormone signaling and actually chronic inflammation. So sexual dimorphism in cancer is something which is quite well known. This is a review we wrote a few years ago. There are others on the topic, showing that many cancer types, the incidence of cancer is significantly higher in the male versus female population throughout in many parts of the world. So many cancer types, there's incidence, also mortality and survival. are different between the two sexes, and with male population doing worse than the female, with the only notable exception of thyroid cancer, which is the opposite for reasons that are not very clear. Now, in the skin, it's probably the best data actually related to melanoma development, where I'm not sure there are conflicting reports about incidence, but certainly in terms of survival, male populations, indicated here by the blue line, the male population does worse than the female population across the ages. So even young boys, supposedly, according to these papers, are doing worse than young girls. So there is a growing interest in trying to understand, establish some kind of a molecular basis for this different susceptibility. These are just some papers, but a number of the others are coming out, notably from Paul Boutros' laboratory in the United States. And pointing to the fact that cancer, so these are using large data sets, and pointing to the fact that cancer developing in male patients, they have a higher mutation rate, and also specific mutation signature, which are different between the two sexes, including also mutations that affect the p53 network. And the other important thing is that these differences appear to be also cancer type specific. So these are interesting clinical papers, and of course what would be important for somebody like me is try to get some kind of mechanistic insights into these differences and try to give a logic explanation of what we are observing. And so our working hypothesis is that there is an important interplay between sex chromosomes and sex hormones, actually a crosstalk between the two of them. I'll be talking about the androgen receptor, which actually is actually mapping in the X chromosome, among other things. And so this interplay between sex hormones and sex chromosomes is obviously playing an important role, both at the level of cancer or cells of origin, cancer stromal cells, and field cancerization is something that to this audience probably does not need to be one does not need to say much about the importance of this in the skin. And also in terms of systemic differences, well known are the differences in the immune system between male and female, and also in metabolism. I like some kind of thinking out of the box. And so this is actually an opinion article that I wrote last year, arguing that actually there is a continuum and also between the two sexes to some extent, and also an interplay and an close interconnection more than we would like to think about gender and sex. And then, so I just refer you to this article, pointing out that another important difference between the sexes between individuals, I say, but is actually also at the level of brain development, which is under close sex hormonal control. So for experimental purposes, we actually been focusing, so we are molecular biologists or cell biologists, and so we are just focusing on cells and tissue, but in a very idealized fashion, just simply studying cells by themselves, epithelial cells, or you'll see also melanoma and melanocytes, and underlying stromal cells, fibroblasts. And obviously the interplay that these two cell types, the crosstalk between the two cell type, and also the immune system. And so now we keep working on this, but actually bringing into the equation also the possible differences between the two sexes. And as a basic biological process that we've been studying is the one of cellular senescence, when the cells exhaust their proliferative potential. So cellular senescence at the level of cancer-initiating cells or cancer cells is thought to be beneficial, because obviously if you exhaust the proliferative potential, you can also lead to decreased tumor formation, decreased risk of tumor formation. At the level of stromal cells, the same process at the same time that is causing a decrease in proliferative potential is actually results in increased production of a number of molecules, inflammatory cytokines and metalloproteases, for instance, matrix metalloproteases, that actually have a tumor-promoting function. So senescence is a double-edged sword on the stroma versus the cells, cancer, the epithelial tissue, and you'll see also in melanoma. So this is another view of this whole process, thinking of the skin now as a field, as a large area. That's why I mentioned we are very much interested in field cancerization, the idea of multiplicity of lesions, and some of them recurrent lesions, and the close interplay between the two compartments of the skin with analogies to developmental signaling, developmental fields like you have been classically studied in developmental systems like in Drosophila. And so for the last few years, we focused on we actually did a lot of work in focusing on the stromal compartment and studying the early steps of conversion of a normal dermal fibroblast into a cancer-associated fibroblast. And focusing on the interplay, the conversion of this dermal fibroblast into senescence dermal fibroblast and the molecular underpinning. More recently, and that's the work was just accepted for publication, we actually now looking also at the conversion and the further evolution of the senescence stromal cells, incipient cancer-associated fibroblast, into cancer-associated fibroblast, with this involving genomic instability and changes in chromosomes. So we are molecular biologists, so we are studying crosstalk between cells, and we are focusing on this particular signaling pathway, which is the Notch signaling pathway, which is an important way of cells, one cell type communicating with the other. So I will skip all the molecular part, just simply to say that there are two key components. One is the receptor, the membrane, which then travels to the nucleus when it's activated by the ligands on neighboring cells. And then there is the second key component is a DNA binding protein, which is called CSL. And so these two molecules are part of the same pathway, but each of them can actually also play some independent or separate functions. And so a recent paper last year, we found that the second component, the CSL DNA binding protein, besides regulating transcription, as is classically known, also binds to telomeres. And it actually is important, it binds directly also to other telomere protective binding and protective proteins, and is actually important for maintenance of stability of telomeres. Chronic exposure to UVA, we show, is actually causing a decreased expression of this CSL, which protects telomeres, and the end result is actually chromosomal instability. And with UVA, as many people know, it actually can directly target stromal cells, not only epithelial cells. So this is actually work done in close collaboration with clinicians. So I'm actually a big fan of interaction collaboration with clinicians. And so this is work done with Vic Neal at Massachusetts General Hospital and Mitch Levesque and Rainer Dummer here in Zurich. And the nice thing about the skin is actually we can obtain and study both tumors, squamous cell carcinoma. I have a problem with this thing. And the surrounding normal skin from which we can also extract cells. And so we then compare the cancer-associated fibroblasts that we extract from the fibroblast, voila, with the fibroblast around it. And so to find that in the fibroblasts that around from squamous cell carcinoma, there are significant changes in telomeres, loss of telomeres, and also chromosomal fusions. We went on and asked whether this chromosomal instability in the cancer-associated fibroblasts is associated with specific genetic alterations, and found that actually the Notch1 gene, that is the first component I mentioned of this pathway we are studying, Notch CSL, we found that the Notch1 gene is actually frequently amplified in this cancer-associated fibroblast. This is FISH, in situ fluorescence hybridization, and the red dots are the copies of this Notch1 gene in cancer-associated fibroblast, finding in a fraction of cells there are 3 copies, 4 copies, or 5 copies of gene duplications. And so this was actually an analysis done looking at matched fibroblast and CAF from the same patients, compared with entirely normal fibroblast, again human. And so the other interesting thing, again, we found an increase, a substantial fraction. These are heterogeneous populations. So within the same calves, we find cells with 2, 3, 4, or 5 copies of this Notch1 gene. In the matched fibroblasts from the same patient, so coming from the dog ears over here, we already find some Notch1 amplification, which is in keeping with the notion of field cancerization and the fact that actually apparently normal skin already contains alterations. This has been very nicely reported by others at the level of epithelial cells. This indicates that even in the stromal cells, we might already have genetic changes. I mentioned that both Notch and CSL can do something else besides working with each other in this Notch signaling pathway. And so work by Fabrizio Dada Garfagna some time ago showed that the Notch1 protein, besides the classical pathway, can also prevent the DNA damage response pathway by directly binding to the ATM, ATR kinases and suppressing all these downstream events that is triggered by DNA damage or genomic instability. As I said, these calves are genomic unstable. And so if you actually look at a calf and you've stained them with a marker of early steps of the response to genomic instability, this phosphorylated histone, you find strong expression of these markers, but all the other markers of the DNA damage response are all negative. If you now take the same calves and you actually abrogate genetically this Notch1 gene, you can appreciate that there is a very nice induction of all this downstream pathway, including P21 and including actual induction of growth arrest. And so this is a paper we just, I think will come out shortly, showing that in the second steps, the transition from a senescent fibroblast to a cancer-associated fibroblast, there is a frequent amplification of the Notch1 gene, and this is actually suppressing this DNA damage response. That follows that if you actually genetically target or pharmacologically target this function or activation of this Notch1 protein, this might have some beneficial effects in retarding or slowing down skin cancer expansion. And so actually this is, we have tested that. We have a model in the laboratory where we actually inject in the ears of mice skin squamous cell carcinoma plus minus cancer-associated fibroblast. And in this case, this was actually injecting mice with the same mixture or combination of cells. But on the right side here in red, the mice were then topically treated with an inhibitor on Notch1 activation, which is a gamma secretase inhibitor, which is widely used. And so you can appreciate that in the topically treated mouse ears, there is a significant slowing down of this tumor proliferation. So this is actually, again, that's why we actually are quite interested also in the translational implication of our work, and this is an example. Now I'll come to the second part of the talk, which again is focusing on senescent aging. And so now we are doing a lot of work at the level of androgen receptor signaling. It's well known that levels of androgens, testosterone in the male population, but also other androgens in the female population, is decreasing with age, which actually is inversely correlated with the increase in cancer risk. So the androgen receptor is a cytoplasmic protein translocates to the nucleus, and actually when it's activated by the ligand, it can actually positively control its own expression. So by looking at the expression of the receptor, you can actually get an idea of the activity of the pathway. This is not the whole story, and this is actually important for the second part of what I'm going to tell you, because when the pathway is activated, the receptor is also downregulated. So in collaboration with Mitch Levesque and Rainer Dummer, as well as Vic Neal, we actually look at premalignant lesions, actinic keratosis in this case, and by quantitative immunofluorescence, by topographical analysis of large section of the skin, for instance, in this case affected by actinic keratosis, we compare level of this androgen receptor in the fibroblast flanking versus underlying or mixed with the lesion, and you can appreciate that there is a decreased expression of this receptor. This is in a dysplastic nevus, and this again in the fibroblast adjacent to the melanocytic lesion, there is a decreased expression of androgen receptor. Is actually androgen receptor important, signaling important in fibroblast or in the conversion of fibroblast to cancer-associated fibroblast. This is a genetic experiment taking entirely normal foreskin-derived human fibroblast, and silencing the AR gene, and in this case, it's looking at a couple of markers of cancer-associated fibroblast. IL-6 is an important pro-inflammatory cytokine, as everybody knows. Periostin is a cancer-associated matrix protein, also very important. And the metalloproteases, for instance, are also induced when we remove or silence this AR. Not shown here, at the same time that we're inducing all these markers of cancer-associated fibroblasts, we also induce senescence, because P21, a classical mediator of senescence, is also a direct target. So there is a technique, a biochemical technique that can be employed to look and study interaction between proteins, which is called proximity ligation assay. I will skip the detail of what this technique is about, but basically you have two antibodies, one targeting one protein, one targeting the other, and then by a PCR-based reaction, you can actually identify, test whether there are some complexes formed by the two proteins of interest. And so you can see that we actually find the binding association, I should say, of androgen receptor with CSL that I mentioned before in a number of cells. This is actually, of course, validated both in fibroblast and cancer-associated fibroblast. This is significance in vivo because like when we perturb CSL expression, when we reduce androgen receptor expression in the fibroblast and mix them with squamous cell cancer, there is an increased formation of tumors, and the same happens when we mix the same fibroblast with reduced expression with melanoma cells. There is again a pronounced increase in growth of these cells. And so the conclusion of this work that was already a couple of years ago is that actually receptor is actually an intrinsic part of the signaling network that is controlling on one end senescence of fibroblasts, at the other end, or at the same time, activation of these cells, which has important, again, translational implication because, as you all know, the androgen receptor is a well-known target of a number of drugs that are used in the clinic. And so you can actually think again of androgen signaling as actually both targeting androgen signaling as both beneficial and detrimental. So at the level of the epithelium, you can argue that targeting androgen signaling in epithelium will actually be beneficial. It will just suppress proliferation of cells. This is obviously androgen deprivation therapy is a mainstay treatment of metastatic prostate cancer. Just to say that there are papers of people working on prostate cancer saying that even in the prostate cancer cells themselves, suppression of androgen signaling is beneficial because it's suppressing proliferation, but it actually might be detrimental because it's increasing cell motility, spreading possibly metastasis. We argue that targeting androgen signaling, again, can actually be detrimental if this is affecting stromal cells, because in this case, by inducing senescence, it can also induce this program of CAF activation. Since we work on skin, we actually wanted to expand our work on androgen signaling to actually look at cancer cells. And so we jump into melanoma, of which I'm really not an expert, but it's so fun to try new things in life. But as I mentioned at the beginning, it's very well known that there is an important sexual dimorphism in melanoma development and progression. And so we asked whether, by any chance, androgen receptor signaling was also important in the melanoma cells themselves and not only in the stromal cells. Now, a possible role of androgen signaling in explaining this sexual dimorphism the idea has been around for a long time. For instance, there was a paper in Lancet like in the 80s. But experimental genetic evidence is actually lacking. So the obvious thing first to check whether androgen receptor is expressed in melanoma cells and whether that correlates with type of lesions or also in the male versus female population. We could find, and so each of these dots is actually expression of androgen receptor in individual cells in these lesions, or each of these dots over here is actually individual tumors. And so you can appreciate that there is a heterogeneity of expression both within lesions and among tumors, but with no consistent trend. And this is actually pointing to one thing I already mentioned at the very beginning, that expression of a transcription factor can be very deceiving, because on one end is an indication of increased or decreased signaling, activation of the pathway, but on the other end can also be that when you have a decreased expression of a transcription factor, it's because the pathway is overactivated. So these expression studies are interesting but not conclusive. Actually, in culture cells, you can find the same thing. This is heterogeneity of AR expression in a panel of melanoma cells, primary cell lines also provided by Mitch. And on the other end, on primary melanocytes, is actually much less expressed. But functionally, if we actually now suppress androgen signaling in a panel of, these are all melanoma cell lines, some with high expression, some with low expression. This is measuring proliferation, cell density, and over here is measuring senescence. And so you can see that all these cells, the green means they proliferate less, so all of them, they uniformly proliferate less when we silence the gene, and all of them undergo senescence with different silencing vector. There is an exciting new technology now that does not depend on classical gene silencing, which is a CRISPR-mediated transcription interference that also is inducing very pronouncedly senescence and suppressing proliferation. This not only you inhibit the pathway and you suppress melanocyte proliferation, this is also done pharmacologically. But you actually can induce increased proliferation of the melanocytes as well as melanoma cells when treated with the ligand. To get mechanistic insights, sorry, am I doing okay?
It's good, yeah.
When we actually, to get mechanistic insights into this aspect, into this function of androgen signaling in melanoma cells, we performed transcriptomic analysis in 3 different melanoma lines, and establishing a signature of androgen receptor responsive genes. And finding that, and this is now going to the data set, TGCH data set, looking at a large panel of melanoma lines and their survival, patient survival, and finding that melanoma which have a signature of genes that is decreased by removal of AR signaling, these melanoma patients, they do better than the one where they have a signature of increased AR signaling. Then further bioinformatic analysis showed that there is an association in this tumor and the profiles that we obtained with an increased activation on one end of DNA damage and DNA repair, and on the other end of increased expression and activity of inflammatory cytokines and STING activation. And so mechanistically, we actually find that in these melanoma cells, when we remove androgen signaling or pharmacologically treat with an AR inhibitor, there is a massive induction of double-strand DNA breakage that leaks into the cytoplasm, shown by these red dots. And at the same time, there's activation of the STING pathway, which is sensing DNA damage. And so this is the green dots over here of the STING protein, which is aggregates and increasingly expressed. I'll skip that for the reason of time, because it gives a mechanism of what we found. Just simply to say that even in vivo, now we have this in vivo ear tumorigenicity assay where we inject now melanoma cells, and these again these are mice treated with the AR inhibitor versus untreated. So there is decreased proliferation of these melanoma cells, decreased tumor size, and at the same time there is increased DNA damage and leakage in the cytoplasm. Increased STING activation by these green dots, and increase actually macrophage inflammation. And in the mouse, we then also use an immunocompetent mouse model with a mouse melanoma line in collaboration with Pinchi Ho in Lausanne at the Ludwig Cancer Center, which shows that actually when we perturb AR signaling in melanoma cells, we also are infecting T cells in a favorable manner with a decrease of Tregs and activation of T killer cells. So this is also a paper that is just coming out shortly, showing that androgen receptor is also playing an important role in the melanoma, not only in the stroma. On one end is controlling proliferation, so if we abrogate the androgen signaling, we actually suppress proliferation and induce cellular senescence. And at the same time, it's actually promoting DNA damage, and therefore it's releasing DNA damage activating STING pathway. For the future, I think it's important to, again, put this in the context of this sexual dimorphism, the interplay between different sex hormones. And so I talk a lot about androgen signaling. There is work in the literature pointing to a possible role of estrogen receptor in melanoma control. That was nicely reviewed by Reinhard. There is a work by Rekeke in Philadelphia pointing in this direction. And it's also important to keep, this is actually a couple of opinion articles that we wrote together with Bernard Demir. Keep important that, for instance, anti-cancer immunity, to keep important the variables of sex hormones levels and also the sexes. And so I find we're just acknowledging people in the work that did the work. So Atul Katarkar and Julia Bottone have been very important for the study that I showed on Notch1 in cancer-associated fibroblasts. And Sumitra Ghosh and Minma in Lausanne have been very important for their, have been leading the project on melanoma and androgen signaling. Thank you so much. I apologize for the long talk, I guess.
No problem. Thank you so much, Paolo. That was a really fascinating talk. I myself was not aware that the androgen pathway plays such a prominent role, at least in mouse models of melanoma. That's very interesting. Maybe also as a future therapy modality, I don't know, maybe Christian, could you tell me, are there some questions that came in online?
So far, no questions online from the audience.
So far, no question. Then my question is, are you really planning to implement such therapy in the clinics? I mean, to treat males with anti-male hormone therapies, of course, problematic, but like in women,
yes. So I would
be
not
much problem.
Yeah, sure. As I said, these are basic studies. You could think, and actually, so in the work we have done, it looks like in the it's dominant the effect on melanoma relative to stromal cells. Mm-hmm. We would like to think that it could be used as a coadjuvant for other therapy. And so we've been talking about Reinhard and other clinicians. This is not what we do, but will be a really very interesting and important application. But
the data looks actually really clear, so
we'll see. I mean, that's what we got. I mean, we certainly tested
yes.
many melanoma lines, and with, as I mentioned, high and low level of AR. So I don't think the expression is, but certainly that actually would, I hope, some clinician will be interested in following up on this.
Well, you have at least already interested me, but Reinhard is the man to talk to when it comes to melanoma.
Yes.
So, well, thank you again very much for coming here and giving a fascinating talk.
Wir haben noch eine Frage
sorry, there's a question. Yes.
Aus dem Saal, Professor Cozio.
Thank you very much for this interesting talk. I have two questions. One is, if I remember correctly, I think androgen receptor is coded on an X chromosome. Yes. And there are 45X Turner syndrome patients, but there are also 45XXY Klinefelter syndrome patients. Do you see a difference? The first question is, do you see a difference in tumor incidence? Is there a gene dosage effect maybe? And the second question is, I think there are mutations also in androgen receptors, complete androgen insensitivity syndrome, CAIS, and partial androgen insensitivity syndrome, PIES.
Okay, if I can rephrase the question just to make sure I understood, because so you rightly indicated, and I mentioned that the androgen receptor is linked to it maps in the X chromosome, and so my understanding of your question whether we look at patients with a different dosage.
Yes, because there are Klinefelter, for instance, which are XXY.
So this is something very interesting that we would like to look at, and so maybe this is this big audience. If somebody is interested, please contact us. We are actually presently looking at the interplay between the X and the Y chromosomes, and so there are 2 or 3 papers that have been published showing implicating some Y-linked chromosome in controlling of AR expression of activity on the X chromosome. fascinating. I don't know if the Y chromosome is controlling androgen signaling, but we are looking into that. And concerning the androgen sensitivity syndrome, for other reasons as well, we have fibroblasts in collaboration with a genetic center that is actually a fibroblast from these patients. We had some interesting results. The only problem is that we have no controls. As you know, each person is different from the other, and also when we get these cells, we don't know how many passages. So all I presented are very early passage strains of cells that we make ourselves. So I actually have a right control. This is a very interesting topic, but yeah, we lack the rigorous setting to actually address an important question.
Thank you. Thank you.
Well, thank you very much, Paolo. Actually, these are some of the best chocolates you can buy in Zurich. It's for your trip back home, so you don't leave hungry. Okay.